So
Zoom is just, half a mile from here, so we can call them up and ask. This guy who started Zoom has done very well.
yeah, certainly.
Whatever happened to Skype? I mean, I used Skype for some reason to Skype the other day, and it was just awful.
Skype is terrible.
Yeah, I mean,
it's always been terrible.
In fact, it was with some people in Israel, and it was terrible. I was, I remember that, and Zoom is just, half a mile.
So, Steve, if you can stop sharing now, because I have an introductory slide to show.
Okay. Stop share.
Okay, good. I'll show this slide then.
Shall we let people in now?
Yeah, I guess we should let all the people in. Let's see. Admit all.
I'm coming during the holiday here. I'm sorry. Hi, Ken.
Okay.
I see Ken Dill there.
Hi, everybody.
Hi, Ken.
Good to see you all.
Okay.
Hello, hello.
Hi, Sue.
Good morning, Steve.
How are you?
I'm okay, just waking up like you.
Yeah, I understand. I got up at an unusual hour.
Yeah, at least we have a little rain here, which is nice.
Not much. It's not. It's it ain't going to change things, but apparently it's going to rain more later in the week, so that would be good. So.
Hi, Steve. Yeah, Shaul.
Hey, Shaul, how are you?
Good, good. Good to see you. Yeah.
Hi, Shaul. How are you?
Oh, hi. Shalom. Yeah.
Good to see you. You're quite a stable presence in our webinar. It's nice.
Okay. Yeah, sure. It's very yeah.
Bill Eaton, I see
Steve,
there.
you are complaining about the early hour, right? Yeah.
Not really.
Steve.
Yeah? Who am I talking to?
That was Shaul.
Steve,
Oh,
this
that's
is Shaul. You are complaining about the early hour.
Yes.
The Nobel Prize is worse.
The Nobel Prize is worse.
The hour for the Nobel Prize. Yeah.
It's not my problem. At least not this year.
Right, yeah.
I see Bill Eaton. Hi, Bill.
Hello, hello.
Hello.
I seem to have a problem.
This is crazy.
Okay. Yeah, okay. So let's wait 1 more minute because the numbers are still growing.
Hi, Peter.
Good to see you.
I don't see you, but that's okay.
Oh, there, now you can see me if you, yeah, great.
Oh, there he is, yeah.
Yeah,
Hi,
good
Peter.
to hear. Yeah, it's a nice topic.
It's kind of an important one.
Oh yeah, I was just giving, well, not just, a few weeks ago, I taught a new course on chemical kinetics and had a part on enzyme catalysis with, and sort of told the history of the Vorschel doctrine. So, which I think is basically correct. But I always tell people about the story that in 77, when I was in Göttingen, he got off the plane and visited us, Klaus and me and Zan, and he said, "I've solved it all."
Yeah.
And that was his electrostatics thing, and he did, but it was interesting.
I will
somewhat
talk, I'll
skeptical
talk
at the
about
time.
that. I'll
We were
talk about
somewhat
exactly.
skeptical at the time.
Well, I can assure you they still are. The
Okay,
but I hope we've put a little bit of a nail in that one. I mean,
yeah, I think it's a
in his
bigger
favor.
part. It's a bigger part.
In his
Although it's
favor.
interesting when you give a lecture for our class, you sort of think about the whole thing,
Yeah.
and there still are quite a few really interesting questions in all of this business.
No,
So, yeah.
Okay, so I guess we're going to start the lecture now. So welcome everybody. Welcome to the Protein Folding and Dynamics webinar. It's our bi-weekly meeting, and I welcome you all. Before I introduce the speaker of today, I would like to tell you a little bit about the way we run the meeting. We try to make it as interactive as possible. Those of you who are frequent visitors to this meeting know it already. We try to let everybody ask questions in the end. But during the talk itself, we would like to ask you all to mute your microphone so that there will not be any noises. In the end, if you have a question, please write it in the chat, and I will ask you to unmute yourself one by one and ask your question. And we will go as long as there are questions and try to answer them all. One more thing that I want to mention is that I'm going to lock the lecture a few minutes after we start, because we've had some disruptions in a couple of meetings ago when we had Zoom bombers entering the talk, and we tried to avoid that. And finally, in 2 weeks' time, we're going to have Michael Woodside from the University of Alberta, and so we welcome all of you to join us in 2 weeks as well. So for today's speaker, it's a real pleasure to have with us today Steve Boxer from the Stanford University. Steve does not really need introduction because he's very well known, but nevertheless, a few words about Steve. He started his career at the University of Chicago, working with the legendary Gerhard Kloss, and then he moved directly after PhD to Stanford and has been doing seminal work in multiple fields ever since. He has made contributions to all the important, I think almost all the important fields in biophysics, starting from photosynthetic reaction centers, then heme proteins, then membranes, where he really made very beautiful contributions to our understanding of supported lipid bilayers. And more recently, perhaps also Stark spectroscopy and the effect of electrostatics in proteins. When I looked on Steve's very long list of papers, I noticed that he keeps coming back to some of these topics again and again and making new contributions, new methods, new ideas, and it's quite impressive. Steve, of course, won many, many awards over the years. He received the Errol Plyler Prize of the American Physical Society. He received the E. Bright Wilson Award of the American Chemical Society. He's a fellow of the Biophysical Society, the American Academy of Arts and Sciences. He's also a member of the National Academy of Sciences of the US and many, many others. And today he's going to tell us about his work on electrostatic contributions in enzyme catalysis. So please, Steve, I will stop sharing now and let you start the talk.
Okay, well, thank you, Gilad. We've been having some trouble with the presentation, so I'm going to stick with this format and hope that it works. Can everybody see this?
Yeah, it looks like
yeah?
It's
okay.
Yeah, it's okay.
That's how we're going to do it. We're just going to do it this way rather than in full screen. So thanks, Gilad, for organizing this. This is a great idea and a great contribution to keep us all busy, though some of us are incredibly busy keeping things going on. I hope everybody's in good health. It looks like everybody I've talked to so far is, and I also wish everybody a happy holiday. So I think, looking over the topics that have been and some of the talks that I've looked at, folding is obviously the headline topic here and is obviously important, but of course, proteins fold to create function, and one of the most important functions is catalysis, and that's what I'm going to talk about today. That's, as for those of you who know me, that's a relatively new area for my group. It's something we've been working on really over the last 5 or 6 years. And it was a spin-off of many other things. And I'll sort of hint at what some of the origins were as I go through this. I'll talk about some of this. So I've got my leading slide here with an outline of what I'll talk about today. I'll talk, and I hope, can you see my arrow here? Okay, is that going to work? Yeah, since we're not doing this in full screen mode. I'll give a little bit of background, sort of following up on what Peter was talking about briefly. Then I'll plunge into what really was the core effort from our lab, which is the development of vibrational Stark effect, which is a really simple experiment, and then an even more simple idea, which is the vibrational solvatochromism, which has many analogies with things that are probably familiar to people from earlier work with electronic transitions. And then using simulations, and I'll say more about that later, trying to get information on absolute fields. And I'll try to explain why the absolute electric fields are important. In fact, why we think electric fields are an ideal language to use to describe these intermolecular interactions, non-covalent intermolecular interactions. Then I'll go through the one example that we've worked on in greatest detail, which is a relatively simple enzyme, Ketosteroid isomerase, or KSI. And it turns out that for a number of reasons, which will become clear, this was an almost completely fortuitous choice, but a really lucky one because everything is simple. And that's not the case in other systems. I could then talk about many other things which are much more recent, but I would focus on just two. One has to do with the possibility of mapping fields and getting more than one projection of the field, and I'll sort of just very briefly speak about that. These last two topics will be very, very short, and then some very recent work connecting this work in catalysis to other areas of interest having to do with bond rotation or bond selective rotation using GFP as a model system. So this is a pretty big menu, and as I say, I'll go through these early topics in some detail, and then the later ones very, very superficially. So this picture that I'm showing here on the left from a famous paper by Wolfenden now 25 years ago makes the point, and it's just sort of in the folding community, one talks about, what's the probability of folding when it seems like it should never happen, and there are many, the problems that are solved involve many orders of magnitude. Well, the same thing is true for catalysis. The uncatalyzed reactions that are involved in these are a number of enzymes up here. I don't even remember what they all stand for. These are many order of magnitude rate accelerations, and of course it's upon those rate accelerations that we are what we are. We depend on that, and we'll be comparing all this catalyzed reactions in an enzyme active site with the uncatalyzed reaction using the same mechanism. This may seem like an obvious thing, but it's often a source of confusion in the field if the mechanism that's used as the comparison is not actually the same mechanism. We'll typically be talking about reactions in water by comparison with the enzyme active site. And sort of the freshman chemistry view of a simple reaction coordinate is we go from reactants to products through a transition state. I'll use that language. I know some people here would prefer to use different language, but I'll use that language because it's the language that's pretty much exclusively used in the catalysis field. And we speak of the catalyzed reaction as lowering the activation free energy. And so the question is really a simple question, but it's a completely general question. It applies not just to biological catalysis, but chemical catalysis as well, which is, of course, a huge business. I was recently told that about 80% of all chemicals are made using catalysts. So this is a very widespread interest. And as I've gotten into this field, I realized that it's very often the case that what is actually responsible for lowering the activation free energy isn't known in almost all reactions. There's lots of speculation on it, but it's not known. In the case of industrial catalysts, people actually don't care. They just want something that works better and cheaper. So the focus of this talk is going to be on electric fields. And when we were talking before, Peter was mentioning Arya Warshall's work. I'll say more about Arya here because his thinking in this area was very much at the heart of our work. This is a super oversimplified picture, but now focusing on electrostatics. Following the last picture, we're going to go from reactant through transition state onto products. And I've made up a system. This isn't a real system. I'll be more specific later, in which the reactant has no dipole associated with it. Transition state involves some separation of charge, and therefore has a dipole. And we'll be interested in the difference between this dipole, the transition state dipole, and the reactant dipole, which in this case, since I've made this zero, is just the transition state dipole. So we'll call this the reaction difference dipole. And I'll be talking about a number of different quantities here. So I want to keep them clear. I'll try to keep my notation clear. So this is just the difference in these two vectors. This is a huge oversimplification of a very complicated problem, but we think that keeping things simple it gets us somewhere. We don't, and other people are doing much more elaborate treatments of this problem. So we're going to focus on these changes in dipole, really on the movement of charge, which is involved during the course of a chemical or biochemical reaction. And if you think about it, almost all chemical reactions involve some movement of charge. And it's often the case that it's an increase in charge going to the transition state. We're not going to talk today, though we can certainly talk afterwards, about motions that bring the system to the transition state. That's, of course, a very interesting and important problem in dynamics. And I'll say a little bit more about it later in the context of electrostatic preorganization. So if we take this simple picture and now focus on how the electric field created by the environment around the reaction might affect this activation energy, we can at least imagine a simple model in which that field and I'm again simplifying my treatment of the field, I'm just going to treat it as a vector here. This is the total field from the protein, and we'll just call it F. That field, if it were properly aligned, it need not be properly aligned, but let's say it were properly aligned with that reaction difference dipole, then it could have the effect of lowering the transition state. By the way, we're using the physics convention here for dipole, the proper definitions. So the signs are all right here. Chemists have this weird opposite sign convention. So it's just a simple dot product in this super simple treatment. So imagine a charge, I just made these numbers up, a charge separated by an angstrom, which is not untypical. And if we had a field, and I'll use units of fields of megavolts per centimeter, you'll see why that unit is convenient in a minute. And let's imagine I had a field of 10 megavolts per centimeter, which as you'll see is a small field created inside matter. After all, matter is made of electrons and protons, so there are very strong local interactions. Then if this dipole of 1 angstrom were stabilized by that field, properly aligned, it would lower the activation free energy by about 10 kilojoules per mole. Now, of course, that goes into the exponent, so that sounds like a relatively small effect, but that could be a fairly large effect. And as we'll see, these fields can be substantially larger than that in organized matter, and therefore this effect can be substantial, okay? Now, in the chemical physics literature, this would be called the matrix electric field, and there's actually a long literature going back many, many years thinking about matrix electric fields, and of course, there's a large body of literature on going back to Onsager and others on thinking about the reaction field. And again, I'll have more to say about that later on. Now, in the context of our problem here, the question is, okay, this is easy to draw a picture like this. The question is, how large is this electrostatic contribution to catalysis? And that's what I'm just going to summarize this field and call it the electrostatic contribution to catalysis. Well, this concept, of course, has been widely promoted by theorists, in particular by Ariel Warshel, but it's rarely, I would say, even thought about seriously by experimentalists. So if you look at lots of experimentalists, they sort of wave their hands and talk about in vague ways about electrostatics, but then move on to more conventional pictures. And those conventional pictures are summarized here. And this gives you an idea. Peter was talking about, well, back in, actually, you said 1977, I think. Well, here's Aria's classic paper, or an extract from it in 1978, in which he proposed that and I've highlighted a few things here, that enzymes can stabilize varied constellations of charges by fixing their dipoles in appropriate orientations. And then going down further, the effective charge stabilization is shown to account for most of the difference between the activation energy of the enzymatic reaction and the reaction of the corresponding model compound in water. And I've shown on the left here, now using kilocalories per mole, because again, we're trying to communicate with the biochemists who tend to use these units. Here's an activation free energy for an uncatalyzed reaction, let's say 22 kilocalories per mole. This happens to be the number that's appropriate for ketosteroid isomerase, and the activation energy in the enzyme is about 11.5 kilocalories per mole. And Aria's view then is that the entire source of this, or most of it, to use his word, is due to this electrostatic effect, the thing that I showed a picture of a minute ago. But if you look in textbooks, that's not what you'll see, even to this day. And it's largely because there are very few experiments out there that actually directly measure this. Instead, what you'll see are really the picture coming from Bill Jenks, who is a very important mechanistic enzymologist coming from a biochemical perspective, and his descendants, and there are many. They're often reviewers of our papers, so we've gotten to know them very well. What Jenks says, and again, this is just an extract from a Jenks paper, is the rate is really, he really thinks about things in entropic terms. It's bringing things together, orienting components, increasing what he calls the effective molarity of one group, and this is in the context of a bimolecular reaction. And there's obviously an important contribution. I mean, that's what organization is all about. The question is really, what is the consequence of organization? Because Aria's view would be that it is organization, but it's organization of charge and dipoles and polar components in the vicinity of the reaction that matters. So this is really, I've sort of made it here into a question, and it is a controversial thing. I can tell you, I had no idea getting into this just how much controversy there is associated with this, because Aria's view is definitely not the majority view among classical biochemists. And so if you pick up a biochemistry textbook, you'll read mostly about this. And the problem, of course, is that what's needed is a quantitative way to assess what these contributions are. And that's what we've been interested in achieving. And you have to be careful, because if you look at Aria's Nobel lecture, he goes so far as to say that there are no experiments that can address these questions. And so that, of course, was definitely a challenge that we took very seriously. So the question is, how do we measure fields in proteins? This is a little bit of a stupid cartoon, but it's about my only contribution to this, because these are my hands. Of course, these sort of red, white, and blue electrostatic potential maps are very widely used, coming originally from Barry Honig and others. But we would like to know what the fields are. And we'd like to know them locally, right at the spot or at the bond that's involved in catalysis. And in thinking about this, we realized something obvious, because we've been doing Stark spectroscopy for mostly on electronic transitions for many years. And we sort of thought, I could go through the history of it, but I don't have the time, that maybe we could turn this around, because Stark spectroscopy is spectroscopy in an electric field. Maybe we can use the Stark effect as a way to learn about fields on a local length scale with many approximations. So that then brings me to the heart of what we actually do, the vibrational Stark effect. So when as I said, we've been measuring electronic Stark effects for a number of years in a number of different contexts. And we got into the vibrational Stark effect, again, partly because of a challenge. We were looking at some intervalence charge transfer complexes, inorganic systems, and these often have nitrile substituents, and we wondered about the Stark effect on vibrations. And we were told that these effects were, immeasurably small because we all teach quantum mechanics, and of course, for a harmonic oscillator, the position of the average position of the oscillator in different vibrational levels is the same, so there would be no difference dipole going from one vibrational level to another. But of course, everyone here, I think, understands that real potentials are anharmonic, and to the extent that they are, there is a difference dipole. Now, I'm again using the symbol delta mu as I did for reaction difference dipoles, but here I'm really talking about a spectroscopic difference dipole. So the dipole going from the ground electronic to the excited electronic state, let's say for a charge transfer complex, or in this case, from the ground vibrational level to the first excited vibrational level. So I'm not going to go through any of the details of how we actually measure these things. We're good at this. We know how to do it. Suffice it to say, we build a parallel plate capacitor, apply a defined external electric field, and there are all kinds of complications there. And it interacts with this difference dipole, and we can measure through the analysis of the effect of an external field on an IR spectrum. We can get out information on this difference dipole. So this is the change in dipole going from one vibrational level to another. Usually, the usual units for dipoles are dBi, but we prefer to use, and spectroscopists typically do, what's called the Stark tuning rate, which is equivalent units, but a little bit odd looking, of wave numbers per megavolt per centimeter. If you stare at that for a little bit, vibrational spectroscopists always have wave numbers on their horizontal axis, so at least it's a linear energy unit, and megavolts per centimeter is a unit of electric field. So this says essentially what the sensitivity is of a vibrational frequency in wave numbers to an electric field. Okay, so if I had a Stark tuning rate of 1 in these units, and it was in a field of 1 megavolt per centimeter, it would shift by 1 wave number, which, by the way, is easy to measure in infrared spectrum if the lines are reasonably narrow. So is the unit conversion with dBi, if anybody is interested. But the key is that this is the sensitivity of a vibration to a field. So these things are not huge, but they're easily measured. We've measured it for many different types of vibrational transitions. Most of our work has focused on nitriles and carbonyl groups. In the context of catalysis, carbonyl groups are particularly relevant because there's lots of carbonyl chemistry and biochemistry. And the extinction coefficients for carbonyl groups is good. The Stark tuning rate is around 1 in these units of wave numbers per megavolt per centimeter. Unfortunately, the carbonyl group comes right in the middle of the overwhelmingly intense amide bands from proteins. And so we have to go through some rather elaborate isotopic labeling and different spectroscopy in order to see these transitions, but we've worked that out. Nitriles are great. They're just not functionally interesting in the context of catalysis. They have good extinction coefficients, and they're in a nice region. I'll mention them briefly shortly. The carbon-deuterium bond is also in a very good region. I'll say just a little bit about that later, but it has a relatively small difference dipole, and it's a very weak transition. Now, of course, this is the vector quantity, the direction for a relatively high frequency isolated vibrational transition, so not strongly coupled to other degrees of freedom in the molecule. The direction of that is essentially along the bond axis. So we can have a 100,000 molecular weight protein, but we know exactly what the direction is of the difference dipole of our probe just by X-ray crystallography. And we've shown that it is in fact along the bond axis by a kind of linear dichroism measurement. We've also shown, I don't want to go into this in any detail, that the difference polarizability, unlike for electronic transitions where polarizability changes can be very large, for vibrational transitions, they are very, very small and can usually be neglected. Now, nitriles don't occur very much in nature, but they can be delivered into proteins on using the technique of amber suppression or other methods for modifying proteins. And of course, carbonyls are often on substrates, and I'll say much more about that. Now, just as a general, since I think this is a pretty general audience, why are we using vibrations at all? People have been looking at electronic transitions and for a long time. The problem is that they're associated with large molecules. And that's my background. So we've done lots of work in the photosynthesis area and GFP, chromophore, things like that, flavins, retinal, these are all large chromophores. Whereas the vibration is really, to the extent that it's localized, just localized to 2 atoms. So it's essentially as small of a probe as we can imagine. We've thought a lot about how one could get a single atom as a probe, but haven't come up with a good way. Maybe somebody has an idea. As I said before, the vibrational transition moment and the change in dipole is parallel to the bond axis. So that's very convenient for spectroscopy. Delta alpha is very small. And of course, IR and Raman spectroscopy are very widely used. And they can be made into fancy time-resolved experiments, though I'll be talking today pretty much exclusively about just linear IR and conventional FTIR experiments. So there's 2D IR, visible pump infrared probe, I'll say a bit about that in a second, and stimulated Raman and other techniques. So there's a very large array. One can look on essentially any time scale by IR spectroscopy. The problems, of course, are that sensitivity is not great. We still don't have a way of doing single molecule experiments. It would be wonderful if we could. There is some progress in that area, largely by Wei Min and a few others. But so far, this isn't a routine experiment at all. And then, as I mentioned, there are these terrible issues of background, which afflict us for carbonyl groups. So the strategy then that we're going to use is of using the vibrational Stark effect takes 2 forms. And the first is to use these simply as a spectator probe. And we've published many papers on this, where we're looking really at changes in the field. So we do that first calibration step in a defined external electric field, and then a measurement step where we measure IR Raman frequency shifts of the probe in situ in response to some perturbation. And that perturbation could, in the protein, could be making a mutation, could be a pH change that obviously moves charges around, introducing ligands, folding and unfolding. A number of labs have pursued that, light minus dark difference spectra, and so on. And lots of people are now doing this, mostly looking qualitatively at changes in frequency, not so much going in depth to understand quantitatively what they mean in terms of the fields, but it's still a very useful approach. Now, a key assumption, this is going to be something that maybe there are questions about, is that this magnitude of the difference dipole is an intrinsic property of the oscillator for all relevant fields, important here, and all relevant non-covalent interactions. If one can in a computer make the field so large that this assumption breaks down, but at least in experiments, and we have experimental evidence, which I won't go through, we can pretty much treat this as a fixed quantity once calibrated. Now, just to give you an example without any data, but to show you, this is because we haven't done the experiment yet, but we're very much on the way to doing it. Here's a really complicated system, the photosynthetic reaction center, something we've worked on for a long time. We excite the special pair and you get electron transfer. I think this is how I first met Gilad many years ago when he was a graduate student. And we now can insert nitriles pretty much at will using the technique of amber suppression. Amber suppression is a way of introducing non-canonical amino acids into proteins. In this case, we use ortho-cyanophenylalanine, and using a technique developed by one of the students in my lab that allows us to do this in photosynthetic organisms. This was a 3-year project. I thought it would be simple, but it was quite a a big undertaking published in this journal that we don't usually publish in, we've been able to insert these nitriles pretty much at will throughout the reaction center. Here's an example. We have X-ray structures in this case. And the idea then is if an electron were suddenly to appear on this yellow molecule, well, we would expect a very large shift in this nitrile. And its magnitude and time dependence, which would be determined by time dependent visible pump infrared probe experiments, would inform us on the mechanism as well as on the local screening inside this complex protein. So here's an example of a 100,000 molecular weight integral membrane protein. We can look at a single bond with no problem. So this is work in progress. Next time, hopefully not with a pandemic as an excuse, but next time I'll get to show you some results from those experiments. Now, in the context of catalysis, our problem is more demanding because what we really need to know is the total field from the protein. And as is always the case, it's easier to measure differences than absolute numbers. So how are we going to get to these absolute fields relative to the field in vacuum? And the method we use here is a really classical approach to the problem. I have no idea why this hasn't been more developed, and that's solvatochromism, so the spectral shift in solvents. And probably everyone here at one time or the other has seen the absorption spectrum of a typical dye molecule shifting, going from blue to red as one goes from a non-polar to a polar solvent. And exactly the same thing, and that's due to the difference dipole between the ground and excited electronic state. I've shown you that there is a difference dipole going from the lowest vibrational level to the first vibrational level of a carbonyl group. And so we would, and we expect the same thing, and we see exactly the same thing. So here in hexane, the band is blue shifted and narrow, and as we go to more and more polar solvents, the band shifts to the red, and it also becomes much broader. So this really is classical solvatochromic behavior. And it turns out this works for carbonyl groups, both in hydrogen bonding and non-hydrogen bonding solvents. That's a detail that's important because it's not the case for, it's not so simple for nitriles. Now, how do we interpret these results? Well, we interpret them using simulations. So what we do is we run simulations. We've done this with many, many different force fields. It's actually a really excellent test for force fields. In fact, we're just sending off a paper now, which is exclusively on that topic. What we do is we, just run a simulation, calculate what the field is projected onto the, in this case, the carbonyl group. We're using acetophenone just as a model system, and of course, the solvent is, partially organized, but it's also free to move around, and so we get a distribution of fields. And in hexane, that distribution is relatively narrow. In water, it's relatively broad. We have, no hydrogen bonds, 2 hydrogen bonds, short hydrogen bonds. These are all, of course, fluctuating back and forth. And we get this information out from these simulations. So the kinds of results we get are shown here. We calculate the field, and this is actually done using a fairly sophisticated force field. So this is an using the amoeba force field. This is work we did with Vijay Pandey and Pengyu Ren, who is one of the developers of the amoeba force field a couple of years ago, and we've now elaborated this in other examples. And what we are plotting on this axis is the average value of that field, because again, you get a distribution. We can also plot the distribution, but let's just focus on the average value. And what we see is that the fields range from about -10 megavolts per centimeter. So that's already in hexane, a fairly large field, all the way up to about -70 megavolts per centimeter. These are negative because these are attractive interactions. And I'll say more about that in a second. Now, what's plotted on this axis is just the peak positions from this experiment that any one of you could do this afternoon, or depending on what time it is in this morning. And that's what's plotted on this axis. So this is an experimental axis. This is a computational axis. And what we see is we get a really nice linear fit over a wide range of solvent polarities. I prefer not to use the word solvent polarity, but rather average electric field, including both hydrogen bonded and non-hydrogen bonded solvents. And the nice thing is that the slope of this line has units of wave numbers per megavolt per centimeter. So that's the Stark tuning rate. And that's what we measure independently with the vibrational Stark effect. And we get the same value. And as I said before, the line widths are also captured by the simulation. So this then becomes a simple way of converting between an observed IR frequency and a field. So we observe a frequency and we get out a field from that frequency, if you are buying into this analysis. Here's a really trivial example where we put that acetophenone molecule onto a small peptide. This will be familiar to some of you. This is a split protein ribonuclease S. Here's the S peptide. And with that peptide in solution, we have a field that looks very much like the amino acid in water. We put it into a hydrophobic region in ribonuclease S. The field shifts to much more like hexane. So think of it as going into a nonpolar solvent, but it's not a nonpolar solvent. It's an organized environment. So a very large change in field. Also notice, and this is important and very much to our advantage, that the lines become very narrow. And that's typical of almost all IR and Raman spectra in proteins. So our strategy then is we combine these field frequency maps from vibrational Stark effects and solvatochromism, measure IR spectra, and we get our information on electric fields. Okay, let me talk a little bit now about catalysis specifically. I've been going through methodology, but I think this builds the framework for how we should think about it. So here again is a simple reaction coordinate that I drew at the beginning, but now a little bit more elaborated for the case of a carbonyl group undergoing a reaction at the active site of an enzyme shown here. But let's focus up here for a second. So of course there is a dipolar character to a carbonyl group. So it's my initial picture of that was 0, but now it's real or it's finite, and we're going to imagine an increase in dipole going to the transition state. And then the question is, differentially, is this a larger effect due to the interaction with the electric field from the enzyme active site relative to the Michaelis complex or the ground state? So that's what we're focusing on. And I'll say much more about this in a second. Now, what we're applying this to is this model enzyme ketosteroid isomerase that we've been working on in other contexts for a number of years with Dan Hirschlag's group, who's worked for a long time on this enzyme. And what this enzyme does is, as the name implies, is it binds at the active site to a ketone. So there's our carbonyl group that we'll be interested in. It engages 2 short hydrogen bonds with an aspartic acid and a tyrosine. There's actually a cluster of tyrosines. I'll say a little more about that in a second. And the mechanism, which is not in dispute, which has been extensively studied, this is really a model enzyme, involves a general base, an aspartic acid here, plucking off a proton to create what would be in ordinary chemical system, a rather unstable dienolate intermediate, which then goes on when the proton is moved back over here, and thereby shifting the double bond from here to here. So that's why it's called an isomerase. And it's a ketosteroid isomerase because the substrate is a steroid. It's not a particularly interesting enzyme in terms of its impact on a broad scale in biology, but it's a very nice model system. These 2 short hydrogen bonds, and when I say short, they're about 2.5 angstroms in X-ray structures. I'll show you some in a second. Turn out to be rather critical here. And so the question then is really simple. If we think this model is relevant, then the field should be focused right on that carbonyl group, because if you look closely here, we're developing charge right here, okay, as we go from this reactant to an intermediate through this transition state. And this is, again, mechanistically not at issue. Now we're lucky because there's a very nice inhibitor available, which is 19-nortestosterone. And you can see immediately, if you're a chemist, this is a molecule which can undergo this isomerization reaction, but which engages exactly the same interactions. And we're lucky to have a supply of this from the former governor of the state of California. So we put this Schwarzenegger compound into ketosteroid isomerase, looking at this carbonyl group. And now we have to do this very careful difference spectroscopy, but we've also made the 018 version, just so we can be confident from the isotope effect that what we're seeing is real. And the result was really a big surprise. There's this huge shift to the red for this carbonyl group at the active site of this enzyme. It's also very, very narrow. Remember, when we had red shifts before going to water, the bands became broader and broader, and that's because of inhomogeneous broadening. But here the band is relatively narrow in the protein, but very redshifted, way to the red side of where there are rare fluctuations. When you run the dynamic simulations, there are rare fluctuations to short hydrogen bonds in water, and that's exactly what's going on at the active site of this enzyme. So it's an enzyme, so we have at our disposal a whole bunch of mutants that other people have made and studied, and where we have rate constants measured. And as we make it a worse and worse enzyme, I won't go through the details here of each of these mutations. These are each single site mutations, making it a less and less proficient enzyme. So KCAT is decreasing as we go here. We see a systematic shift to the blue. Okay, and so right away, if you look at this, you'll say, well, there seems to be some relationship between this quantity that we've been identifying with the electric field and KCAT. Now, a legitimate concern, and this is a concern that is common in all of site-directed betagenesis, is that most mutants that we make are rather brutal. If we get rid of a functional group, we might, let's say, introduce a water molecule or some other thing. So we use the technique of amber suppression, which produces smaller perturbations, but nonetheless are systematic on this cluster of tyrosines. I mentioned this tyrosine before, but there are two others which also have short hydrogen bonds and forming a little cluster. I could give a whole talk on that. Tom Marklin's group working with us has done a lot of simulations on this cluster. And what we do is we systematically introduce chlorotyrosine at this tyrosine, or this one, or this one. And if we do it at this one, these are color-coded, the one that's more remote, we get a small shift and a small reduction in KCAT. If we do it at this one, we get a larger reduction in KCAT. We do it at this one, we get the largest reduction. A systematic change. You see how nice it is to have narrow bands. Here's a double mutation. So these are changes, and we have obtained X-ray structures for all this. This was work by Yufan Wu, a graduate student in my lab. We do lots of X-ray crystallography now, and these structures are identical to within the resolution of the structure, which is unfortunately still a limiting factor, because X-ray crystallography rarely gives super high resolution structures, but these are pretty good structures. So now if we take these frequencies and we use our analysis of those frequencies to interpret them as electric fields, plotting that on this axis. So this axis is those IR spectra, but now interpreted through the vibrational Stark effect. And if we plot on this axis the activation free energy, and we're using transition state theory here, we're just using it in a consistent way. And if we, so here it is in wild type, these are the numbers I mentioned at the very beginning, 11.5 kilocalories per mole. So that's the fast enzyme, and we make it worse and worse and worse. And so if we plot that activation free energy barrier versus the electric field, we see a really robust linear correlation over a very large range of electric fields. So this is what a physical organic chemist would call a linear free energy relationship. But unlike any other linear free energy relationship, where usually you have some parameter, a Hammett parameter, some other thing on this axis, this is an absolute number. This is electric field, something you can actually calculate from first principles. So it's a really different in that sense. So if we take that and just simply draw a line through it, the slope of this would give us this difference dipole going to the transition state. But that's really not our main focus. Our real focus is what would happen in imagination, if we shut off the field from the enzyme, that is, what is the y-intercept? And if we look at that, that gives us what the activation free energy would be in the absence of electrostatic. So that's the quantity which is what we're really after. And now plotting it back on that original activation free energy diagram that I started with, here's the wild type enzyme. What we're finding is that the zero field, if you like, imagined activation free energy barrier is 11.4 kilocalories per mole. And so about 70% of delta G double dagger is due to electrostatics. And then the rest is presumably due to what Jenks was talking about. So I think 70% constitutes most, but not all, going back to Aria's language. But the real point here is that we now have a way, in a general way, of quantifying this. It's no longer something we can, that we just have to wave our hands about the way is often done in biochemistry textbooks. So the biochemistry textbooks are basically incomplete, and one has already changed. Hopefully there'll be more changing now as a result of these experiments and acknowledging the predictions from Aria Warshall. Now, there's another aspect to Aria's work, which is electrostatic pre-organization. This is a fairly complicated topic, and now from now on I'll be simplifying everything. Basically, we pulled a fast one here because we treated this as if the field that was relevant was the same in the ground state and in the transition state. But we know from just chemical reasoning, and we've shown it obviously by gas phase calculations, that there should be a substantial geometry change going from one state to the other. And so really, what is the relevant field? Well, in principle, we could deal with that by knowing what the field is at the transition state and the field at the ground state. That would be the correct way to treat this. Whereas here we were doing a simple linear scaling. So we were interested in this, and to make a long story short, what we did was use a combination of X-ray crystallography and calculations. This was published fairly recently in JACS, if you're interested in the details, sort of modeling the enzyme substrate complex with this molecule, and then the enzyme transition state with 19-NT. And what we found by X-ray crystallography, I'll just focus on the structure, is interestingly, the ground state structure is distorted towards the transition state geometry. And so it turns out there is a relatively small geometry change going from the ground state to the transition state. And so we were, this more complicated expression, really, we got away with murder here using this simple expression, largely because the enzyme distorts the substrate, presumably electrostatically, towards the transition state geometry. That's an example of electrostatic pre-organization. So just to summarize this, and I want to then talk just briefly about some other topics. I've been arguing that the vibrational Stark effects are quantitative and directional for fields and proteins. And this is completely general. It can be applied to any system. We use solvatochromism and simulations. Simulations are essential here, but you can turn this around and say, well, this is a really good way to test simulations, because these are very local specific interactions of the kind that we're interested in, rather than bulk properties against which many force fields are compared. In this case, in the case of KSI, we have a direct connection between electric fields and the activation free energy, which gives the electrostatic contribution to catalysis. And this, to summarize this pre-organization story, because I think this is really what matters. This is a question that often comes up. Well, the field in water is actually pretty large, but pre-organization of the electric field is fundamentally different from the solvent reaction field. So that's the field that the solvent has by virtue of being organized by the molecule, by the solute around it in the ground state. So it's optimized for the ground state charge distribution. And ordinarily that would have to reorient, that's the underlying concept of Marcus-Hush theory, to accommodate the transition state as the reaction proceeds. And that's what actually limits the catalytic capacity of water, despite the fact that its average value of the electric field is substantial. This is a really important point that is completely obvious to people who come, let's say, from electron transfer background. And without going into anybody else's results, a whole bunch of groups have jumped in and simulated our results. This is a continuing effort in many groups, Teresa Head-Gordon in particular, Tom Markland in our department, and actually they get results which agree surprisingly well with the fields that we're getting. Of course, they can then dissect from simulations what the origin of the fields is, and it's these short hydrogen bonds that dominate the electric fields. And I would argue these concepts should apply in general to any kind of catalysis, including chemical catalysis. Okay, now just two quick stories, and I'm going to go through them really quickly, even though I could say much more about them, but just to give you a flavor of where we're heading with this. So this is work that's very much preliminary and in progress. It's the work of a graduate student, Chu Zhang, looking at an enzyme, which is, again, a model enzyme extensively studied involved in a hydride transfer reaction. That's liver alcohol dehydrogenase. And we were interested in this initially for two reasons. One is it catalyzes a reversible reaction. And we were interested, so if the transition state difference, the transition state dipole is the same going in the forward and reverse direction, but the dipole moments are different, then delta mu of reaction should be different in the forward and reverse direction. We thought that would be interesting. We think it is interesting. And the second is that this enzyme has a non-catalytic but critical ion, zinc ion, at its active site, which we can change to other ions. What's shown here is the X-ray structure at the active site. This is from a structure that Chu obtained using an inhibitor, which is shown here. And here's that carbonyl group interacting with a critical serine residue and with that zinc ion. Now, what's interesting about this is that this involves the conversion between aldehyde and alcohol. And so we were interested in the aldehyde. And what's interesting about aldehydes is that we have not only a carbonyl group of the kind I've been talking about, but now we have the possibility of also looking, not quite perpendicular to it, but at 120 degrees roughly, the carbon deuterium stretch, okay? And that would give us then two projections of the field, which would ultimately be a much more stringent test for calculations, or for simulations of the field. And just doing the vibrational solvatochromism, there's already a huge surprise, at least to me, though I think it's maybe not too surprising in retrospect. And that is, if we just look at this, this is just acetaldehyde with the deuterium here. We look at the carbonyl group, it just looks like the other carbonyl groups I've shown you. Then hexane, it's blue, and if you go to more and more polar solvents in water, it's redder and broader. But the CD bond, if we look at that vibration, which is a very weak transition, so it's hard to do, and ignore this here, this is some overtone band, this actually shifts the opposite direction in as we increase the solvent polarity. So we're just now working with Tom Markland's group, we're doing extensive simulations on this, and if you use simple molecular dynamics, you actually predict this. And what seems to be going on is that the solvent is organizing around the carbonyl group, stabilizing the carbonyl group, producing these negative stabilizing fields, and that then has the consequence of producing a destabilizing field that's nearly perpendicular to that. And that's a pretty interesting result, because we tend to think about fields that are stabilizing, but fields that are destabilizing can also be important, or they can actually work against you depending on the mechanism. And in fact, if we put this into the protein, you see something which I think is really remarkable, and this is just sort of where we're at right now. As it was the case for KSI, but not so far, this band is shifted, the carbonyl band, way to the red, not as far to the red as it was at the active site of ketosteroid isomerase, but it's a weaker hydrogen bond environment. But the CD bond is shifted way to the blue. So actually, this is deactivating, if you like, this site for catalysis. In any case, I thought I would show you this. It's really an intriguing result. The fields are highly non-isotropic. That won't come as a surprise to anyone. This is, I think, going to be a real excellent test for computational people, and we're working on mutants, metal changes, and simulations. Now, the last thing, if I have just a few minutes, and this is work that was published recently, and it may seem like a completely different subject, but I hope I can just wrap up by giving you a flavor for it. So, and this is a problem a lot of people in Israel actually have worked on this problem historically. And it has to do with photoisomerization. Sounds like a completely unrelated problem, but I'm going to argue that it's exactly, or part of it is exactly the same problem. And let's just look at retinal as an example. So if we take retinal in solution and shine light on it, it undergoes isomerization around each of its double bonds, essentially statistically. You get essentially equal amounts of trans to cis isomerization starting from the all-trans form. We start from the all-trans form in a protein, however, as we all know, and as those of you who are watching this, I see there's Stephen. Hi, Stephen. We get a very specific isomerization around just one bond in your eyes while you're watching this, whereas in I'm sorry, in microbial rhodopsins, you get it around just this one bond, and in animal rhodopsins, just around the 11 double bond. So the question again is, what's the origin of this? And this is sort of steric, so this is a little bit like that other stuff, or maybe it's electrostatics. You know, you say electrostatics, that seems like an odd idea. The natural intuition here is that this is a steric effect. And it is in part, for sure. I don't want to argue that it isn't. We're using GFP as a model system. We've worked a lot on GFP over the years and trying to understand excited state properties of GFP. And let me remind you that the GFP chromophore by itself, shown here on the left, is non-fluorescent. I mean, it essentially has a very, very low fluorescence quantum yield, whereas in the protein, it's reasonably fluorescent. That's why it's useful. And what's going on here in solution is that we have competing non-radiative decay pathways. And you know, just making a very simple picture of this, we could imagine if we excite this nominally planar molecule that we might get a flip around, a ring flip around this phenolate ring here, or rotation around the double bond here around the imidazolinol ring. That's what we would call cis-trans isomerization. Both of these could be competing non-radiative decay pathways in solution, but you can imagine in the protein they may be constrained. Well, where would electrostatics come into this? Well, electrostatics comes in because it has been shown, and again, this motivated very much by calculations, in this case coming from my colleague Todd Martinez, whom we work very closely with on GFP-related problems, is that there's a difference in electron flow, whether we do rotation around the P bond, around the I bond. That's not entirely intuitive, but it comes out of high-level calculations. So once again, we have something very much analogous to what we talked about in catalysis, which is the difference dipole involved for a reaction, only now it's an excited state difference dipole. Okay, so this is really catalysis in the excited state, if you like. Think of it as an excited state enzyme. So the experimental approach, I'm not going to go through any of the details here. I'm just going to summarize this. All of this was published earlier this year in a paper in Science. If you're interested in the details, including extensive supplementary material where lots of the real details are, is we tune both the steric and electrostatic properties of the chromophore or the environment. The environment part of this hasn't been published yet. We're just finishing a paper on that now, but it follows very closely what I'll be saying now. And then systematically study the effect on function. In this case, our function is a simple measure of the energetics associated with isomerization reactions. Now, we've been studying this. There's a long history to why we're interested in studying this process. But all I'm going to talk about today is this step here, which I'll call step 2, following excitation, whether we go over this barrier, which is analogous to what I talked about in catalysis, on to a conical intersection where we can either come back here or on to the to the trans state. So we're going to focus on this, and we get that information on the energetics of this barrier by simply measuring the temperature dependence of the fluorescence lifetime. Now, the trick here is, how do we perturb the system electrostatically? We do it using amber suppression. There's a tyrosine residue in the chromophore, and we can put in almost anything we want here. We put lots and lots and lots, more than were in this paper actually, substituents onto that phenolate ring, which either change things sterically, or we can look at isosteric series, or electrostatically. So we're really doing physical organic chemistry on this very complicated thing. And we can introduce this pretty much at will and essentially in 100% yield using amber suppression. We're using a photo, for those of you who are GFP experts, we're using a photo switchable variant. This is one that's used in super resolution microscopy because it has a good probability in the light of undergoing this cis-trans isomerization. I should mention that the chromophore itself is in the cis form in the ground state. So we have X-ray structures of all of these except for one with monofluoro, and in most cases, interestingly, we see a single orientation, even though we have substituents that could be on either side. These are reasonably high resolution structures. This Drompa is actually a difficult protein to work with. If we look at the absorption shifts, and it shifts in a very systematic way. And again, this is just classical physical organic chemistry, whether we have electron donating or electron withdrawing substituents. So just using a sort of classical Hammett kind of analysis, or what's called resonance color theory analysis, we can rationalize these changes in absorption. We haven't done this quantitatively. Somebody else will almost certainly do this now that we've published these spectra. But you can imagine, stabilizing or destabilizing this transition, so changing the excitation energy by virtue of putting substituents on there. Now, the excited state barrier is what's interesting, and I'm skipping a lot of work here. We measure that by measuring the temperature dependence of the fluorescence lifetime, and what we see is interesting. What's plotted, it's a little complicated. What's plotted here is the barrier height in kilocalories per mole, what we measure experimentally, just again from an Arrhenius dependence of the fluorescence lifetime, as a function of these electron withdrawing or electron donating substituents. What we see is that the wild type is near the peak here, and we go down on both sides, depending on whether we have electron donating or electron withdrawing substituents. We're plotting this against the transition energy, which is, so we have energy versus energy here. So this is a meaningful comparison. And what we see is that the slopes going here and here, we of course only have a fairly small number of points, are about the same but opposite in direction. So this barrier seems to be very sensitive to electrostatics. And as I mentioned, though I don't have it yet in this talk, we have many, many perturbations in the environment which can compensate for or enhance these changes due to substituents on the chromophore itself. So electrostatics must be important. And so the question is, can we connect it with energetics? And now I'm really making a very long story short. I see I'm going over in time here, so I need to do this. Let me go back there. That picture I just showed you briefly is actually what explains everything, but let me just wave my hands here. What we're interested in is whether there's a similar probability of p or I bond rotation. Remember, whether there is depends on the direction that charge is transferring when we make that isomerization process. And then the question is whether we're lowering the barrier here or lowering the barrier here. And our intuition says what an electron donating and electron withdrawing substituent should do. And we are able to, in fact, quantify that, or at least qualitatively, semi-quantitatively, analyze that. And for example, if we have an electron withdrawing substituent, we can show that this biases the isomerization pathway towards this I-twist, which is associated with the movement of charge in one direction. But contrary, when we have an electron donating substituent, it lowers the barrier in the P-twist direction because charge is shifting in the other direction. I know I'm making a very long story short, but that's the essence of it. So it really comes down to this direction of electron flow, exactly the same thing that we were concerned with when we talked about catalysis and delta mu of reactions, okay? So the electrostatic sensitivity stems from this charge transfer during these electronic transitions and the isomerization around these bonds. So it's none other than our friend delta mu reaction. This really provides a generalizable framework to incorporate electrostatic effects into the design of any photoisomerizable system. And then it makes a connection, I think, back to electrostatic catalysis. And ultimately, you know, I think Stephen would appreciate, we always like to say this should also be a design principle for designing catalysts. And there are several labs, including ours, who are now doing that. So going back to the very beginning, I gave some general background, the vibrational Stark effect, and I see Stephen is on the line. I really credit him with the person who, you know, we were doing vibrational Stark effects, and Stephen came into my office one day and said, you know, why don't you do something interesting with this? And catalysis might be an interesting topic. And I didn't know much about catalysis. He knew much more, still does. And that really led then to this work on ketosteroid isomerase. But as I hope I've shown you, we think this is a very general approach. I think this business with looking at two projections of the field will turn out to be a rich area for investigation. And this can even be used to talk about something as complicated or seemingly unrelated as bond isomerization. So here's the cast of characters, many of whom have moved on. Here is Stephen, who's actually on this call, who did this work on catalysis with postdoc Sian Bachi, who came to our group from Robin Hostrasser's lab. Nick Levinson played a very important part in the original vibrational solvatochromism work. He's now at the University of Minnesota. I should mention Stephen is at Johns Hopkins and is no doubt looking for graduate students and postdocs. And so you should send them all to him and not to me, depending on how good they are. Nick came to us from John Currien's lab in Berkeley, and he really, I would say, brought crystallography to our lab, which has now become just a bread and butter part of our work. Yufan Wu followed up on Stephen's work with these chlorotyrosine experiments, and also that recent work on, which took a long time to gestate, on electrostatic preorganization. This is a cast of characters who are working on other enzymes. We are very actively working on the beta-lactamases to try to understand if there's a connection or what the connection is between evolution and these electric fields, if there is one. That's an interesting subject in itself. And then this work on liver alcohol dehydrogenase that I very briefly mentioned from Chu, who's actively pursuing it. And then the last story on GFP electrostatics was done by two graduate students, Chion Lin and Matt Romai. And we're very much indebted to a staff scientist at SSRL, Matthews, who's been actively involved in all aspects of turning us into amateur X-ray crystallographers. And this work has been supported continuously, and I just got good news on a grant renewal by the NSF and the NIH. So Gilad, who as I see there, I remember meeting as a graduate student, I believe, and he's evolved in many interesting directions. Thanks again for inviting me. Sorry for going a little late, but I hope I've given you a flavor for a lot of what we're up to. Thank you.
Well, thank you, Steve, for this beautiful talk. And the floor is now open for questions. And the first question is from France.
Hello, can you hear me?
Yes.
Yes.
Not well. But,
Oh, okay. I'll try to move a little closer.
Yeah.
So, excellent and fantastically presented talk. It's outside of my field of expertise, and I learned a lot. It was good to follow. I have a question about the dehydrogenase story, where you showed fields in two directions changing.
Yeah.
And you showed in some examples how the lines often they change both in position and also in width. And in the example you showed, one of the lines on the carbonyl is actually getting broader, but the CD bond doesn't really see the change in its width. And what was the explanation for that?
Can you hear me? Yeah. It does change some, but you know, you're right. I mean, the difference dipole is smaller, And we're doing, I don't know the answer to your question yet. We will know it pretty soon, however, because we're doing simulations. In order to do simulations on a system like this, or I mentioned beta lactamases, so where we look at penicillin, it turns out that, again, you know, the ketosteroid isomerase example was remarkably fortuitous in many, many ways. That acetophenone data that I showed you early on as our sort of model system behaves just ideally. As soon as you put things, make things a little more complicated around the bond, the simulations get more complicated. And you have to do extensive simulations, and that's what we're doing now on that. And so I'm avoiding your question because I don't know the answer yet. Suffice it to say, let me tell you what the result is for a molecule like penicillin, which has just a simple carbonyl group in a 4-membered beta-lactam ring. It has near it a side chain and a charged side chain. And the combination of those two dramatically change the solvatochromism experienced by the carbonyl group. And through simulations, you can see that, because you can see how locally the solvent reorganizes in response not just to the carbonyl group, but to the other stuff that's around it. I suspect the same thing is going on with that aldehyde, but I don't know yet. We'll know soon.
Thank you.
You know, there's a fellow at the University of Connecticut named Gascon who's a theorist who has done an extensive analysis of using the vibrational start data, interpreted the way we interpret it, as a metric for force field development. Because it's, you know, unlike looking at, you know, a dye molecule, here you're looking at a specific bond. It's a very local probe. And I think this was really the insight from Nick and Stephen originally, working on making this connection through simulation. But actually, it's a really useful tool from a force field perspective.
Okay, next question, Abhilasha Kumari.
Yes, sir. I just wanted to know what is amber suppression and how is it
I'm sorry, I lost you there, but I think I understand the question. So amber suppression is a technique for introducing non-canonical amino acids into proteins. It was a technique that was originally developed by Chamberlain, Hecht, and Pete Schultz. Pete Schultz tends to get most of the credit for it, but others were involved in developing it. And it is a way of changing the codon for an amino acid to the amber codon, and then replacing, and there are all kinds of strategies for how you do this, the aminoacyl synthetase to have the to recognize the amber codon and then artificially aminoacylate that synthetase, originally done by, you know, in a test tube, now done catalytically in the organism. So it's a very powerful sort of, I would say, alternative to or supplement to site-directed mutagenesis. And I think it's fair to say that anyone who's done it, it looks easy, but it's actually not always so easy. But we've gotten pretty good at it. It's gotten much, much easier in the last couple of years due to the work of people like, there's a group, for example, if you're interested in it, there's a group at Oregon State, Ryan Mail, has a big center for the development of the methodology, and they share reagents, and they can be very, very helpful. We actually depend on them for many difficult situations. Does that answer your question?
Yes, sir. Thank you, sir.
Yeah.
Okay, next question is by Bill Eaton. He asked me to read it. He says, Steve, have you thought about doing QMMM simulations to watch the KSI reaction, and provided you can find a good reaction coordinate, possibly learn something from comparing the properties of the reactant and transition states?
Yeah, well, in fact, you know, that's been done. Part of it was done by Stephen working with Tom Markland, and also done by Teresa Head Gordon, and actually several other groups. I don't know, Stephen, you would be a better counter of how many groups have actually done the KSI calculation. I think what really comes out of those calculations, Bill, and it's most useful is that it allows, just like with the simulations of the solvents, by the way, is that you can take, you can look in the simulation at what you can't look at experimentally. Namely, you get snapshots of what the important, interactions are, and you can break things down in ways that are very difficult to do experimentally. And the bottom line is that the dominant contribution to the field is those two short hydrogen bonds that you could see us perturbing. It's a little more tricky, actually, in the case of liver alcohol dehydrogenase to do analogous experiments. But I don't know if that's actually answering your question, but yeah, that's been done, and it's being done now in a number of other enzymes as well. I mean, this is a different way of looking at the problem from the conventional way, but it's really sort of getting to the heart of what is responsible for lowering the activation free energy.
Shaul
Yeah,
Mukammel? Oh, Bill.
I guess I was thinking of actually comparing the dipole moments in the ground and the excited state in the transition state by looking directly at the transition state, which you can never do by experiment, but you can do by simulation.
Yeah, well, in fact, I'm sorry, maybe I misunderstood. Oh, that was exactly what was done in that paper on electrostatic reorganization. I showed you just some snapshots, mostly focusing on structural changes, but there are lots of computational work in there. And I guess I would refer you, Bill, to that paper because it's complicated and it involves details. It was published in JACS, because Stephen's still an author on that paper, a couple of months ago. Okay, and we did exactly what you say, or what you're asking about, comparing things in the gas phase with what goes on in the enzyme, informed by structure.
Okay, next question by Shaul Mukammel.
Hi, Shaul.
Shaul, if you can unmute yourself.
Yes, sorry. Yeah, so thank you. It's a great talk. I wonder if you could comment about a possible time-resolved version of this, like by using single cycle optical pulses. And then in a related issue, can you say something concrete about conical intersections using these Stark measurements? Thank you.
I think I can answer the second question first. And I don't think the answer is probably no. Let me go back to your first question. And again, we've done one experiment that I actually like. It's not gotten much attention, but it preceded some of this work. It turns out you can put molecules at the active site of KSI, which are classical solvatochromic dye molecules, okay? And that upon excitation involve a large displacement of charge just because they're charge transfer charge transfer transitions. And what we did was we put nitriles at various strategic locations around the active site, okay, of KSI, and asked, so these were now visible pump IR probe experiments. These were done by a postdoc, Santosh Kumar, in collaboration with Kelly Gaffney's lab at SLAC. And what we observed was actually very interesting. You know, as soon as you excite the molecule, of course, you get an immediate shift, a Stark shift, due to the change in the dipole moment of the excited state. But then, rather than getting a solvation shift of the kind you might expect in an ordinary simple solvent, so the analog of a dynamic Stokes shift, but now looking at a vibrational relaxation, what you saw was the shifted vibration intensity just simply went down, and it recovered as we went back to the ground state at the frequency of the ground state. So there was no evidence from that for solvent reorganization, if you want to use that language. And we did not just looking at the frequency, but also doing polarization measurements. So looking to see whether the nitriles, in this case, these were modified cysteine groups, modified as thiocyanates, whether they underwent any rotation. And the answer was, within our ability to measure it, no. So this was actually kind of a precursor to this notion of electrostatic pre-organization, which I think, at the time we were looking at it, I'd say rather naively. So I'm not sure, Shal, if that actually addresses your question. Do you want us to look at the reaction itself in real time? Is that what you were driving at?
Yeah, that will be absolutely great that you can
Yes,
watch.
it would.
Yeah.
Yes,
Yeah.
if we had some way to synchronize it. Interestingly, the experiment I just described is kind of a fake version of that, because it turns out that if you look at the way, for this dye molecule that we put in the active site of KSI, the way the charge shifts going to the excited state is not that different from the way charge shifts going to the transition state of KSI. So you can think of it sort of as a transition state analog, if you like, in the excited state. And so we've sort of done a little bit like what you described, but no, I don't know how to make this reaction into something we could study the way you described. If you have an idea, let us know. That would be great. I mean, we have all these fantastic tools available now, but you may be shocked to see that all the experiments I described today used absolutely simple methods. I mean, nothing fancy here, linear IR, but some fairly strategic approaches.
Okay, thank you.
Okay. Peter Walliness has a question.
Yeah, well, first, yes, it's a really, really elegant story you have, and I wanted to sort of return to the big picture questions. First, with a somewhat of a historical remark that people might be amused by, you didn't mention that the basic idea of stabilizing the transition state in enzymes as the explanation of the miracles is due to Pauling, who published it in that famous high-profile journal called C&E News, which is not the normal place to publish your important theoretical contributions. But
Peter, can I interrupt you? Just,
yes.
For your own amusement, and because Stephen is on this call, if you go to my website, you'll see, I think the first paper from Stephen actually was on exactly an analysis or reanalysis of Pauling's ideas. And it's a little more complicated than Pauling, but it's a really complicated story. But if you're interested.
Well, I mean, it's also interesting. Anyway, in fact, that's where I was really going to. That I think one of the things that made the Pauling idea seem maybe over simple was the idea that there'd be only a single transition state in order to carry out most enzymic processes. And that's especially clear when you think about there being, a full energy landscape of possible structures in the protein itself. And I guess one of the questions I have is the ketosteroid isomerase just, you said it's the most one of the most highly characterized examples, but how often is it that you have just one single rate limiting step that has the rate that you have? And how often is it that you actually have to do 5 different transformations in the active site before the reaction's over?
Oh, there are many cases like that. I mean, if you look at the reaction coordinate for many enzymatic or non-enzymatic transformations, they involve multiple transition states. This is again a relatively simple example, and I think it's fair to say we were very, very lucky, okay, in that many things that could have complicated the problem turned out to go our way. Or maybe we made them go our way by being simple-minded about our approach to it.
Well, I'm not being critical at all.
No,
it's nice to zero in on that. I mean, it's really important to get that nailed down. But I guess one of the things that I would say still leaves a little bit of a miracle here is if you have to do 5 different steps, you have to design your protein to make each of them have fairly low variables. So now you have to do a little more than one electric field. It might still be all electrostatics, but it has to be very cleverly set up electrostatics, I think, for those reactions.
I mean, it's a great question, and we have not looked at it. I mean, Peter, we sort of started this. I'm hoping that other labs will pick up on this will pick up on this approach and apply it to more complicated systems. We tend to try to look at the simplest systems just to get the basic picture that other people will understand and then can make.
Yeah, no, it's very elegant, very elegant.
Yeah, there are so many approximations. I mean, Shaul is probably horrified by some of the oversimplifications that we've made here. But it basically captures, I think, most of the story. And we'll leave it to others to make it more elegant.
Okay, I guess I will ask the last questions for today. So you started the last part of the talk with the retinal proteins.
Yeah.
So is electrostatics also important for retinal proteins to the same extent?
As with GFP, we suspect so. Interestingly, again, it's Ariel Warshel who's actually written quite a bit on that topic. Again, many years ago, and the answer is probably yes. But I don't want to overstate the importance of electrostatics for isomerization, because there's also clearly going to be some steric contribution. What's interesting is if you look at these isomerization reactions, and depending on the mechanism that they take, they can be relatively volume conserving. So we published a paper late last year, actually the work of an undergraduate, and I urge anyone interested in this question to look at it. Using amber suppression with that chlorotyrosine, we can now address the question. If we flip over, we could flip one way or the other, depending on what the mechanism is. But with chlorine there, we can actually determine which way we flipped. And it turns out to be the volume conserving way. So if it's volume conserving, then probably sterics are less important, and therefore electrostatics may be more important. We haven't done any experiments on, let's say, bacteriorhodopsin or rhodopsin. I'm very much, I mean, this paper wasn't published that long ago. I'm very much expecting that others people who are experts working with those proteins will do it. GFP is a very simple case, again, somewhat ideal because it's so easy to introduce perturbations by amber suppression. We have a paper which we're just in the process of finishing now, in which so I mentioned very briefly, rather than just looking at substituent effects, which is kind of the classical way of thinking about this, where we make perturbations in the environment in combination with those substituent effects and make predictions about whether they should enhance or compensate for the effects based on electrostatic reasoning. And in fact, in all cases, we get exactly the results as predicted at a qualitative level. It's actually a really nice story, and it kind of explains a lot, I think, about why some of these design efforts have been so troubled. It's because people don't have this underlying picture of the energy landscape, to use Peter's language, of what the reaction is actually doing. And so they're either just giving up and doing directed evolution, or they're trying to design, but I think they're designing the wrong thing. And so we're arguing about what the right thing is that people should be designing. I think they should be designing fields. And I think, several theorists have picked up on that idea and made predictions. So I think that'll turn out to be a rich area for the future.
Yeah. Okay, thank you very much. So I would like to thank all the people who stayed all the way to the end of this really interesting question and answers session. And I would like to thank Steve again for the wonderful talk, and hope to see you all in 1 month, because we have a break for the holidays, but in 1 month with Michael Woodside. Thank you very much, and bye-bye.
Okay, bye-bye, everybody.